All right. Good morning, everyone. I'm Yotaro Sueoka, a PhD student working with Jim Knierim and Noah Cohen, and I'm very excited to be here. Okay. Okay, so this is a spatial navigation section, so it is perhaps needless to say that spatial navigation is important. And from migrating birds to the wildebeest in Tanzania, it is critical for survival to know where you are and where you're heading. So how do animals achieve spatial navigation? Well, one way is through landmark-based navigation, where animals use external cues such as visual landmarks and olfactory cues to know where they are in 2D space, or perhaps 3D space. Another method is path integration. And in path integration, animals use their self-motion cues such as vestibular inputs, proprioceptive inputs, or motor copy. And by integrating these motion signals over time, one can estimate the displacement through space. And path integration is especially important because landmarks are not always available, for example, in darkness or in featureless environments. Take, for example, the Saharan desert ant, which is the world's best path integrator, and they can count their number of steps and estimate the location. Oh,
Can you turn on your Zoom video?
Zoom. Oh, okay. Hmm. Let me stop.
Don't stop sharing.
Let's see.
Okay, it's not important. All right, just trying to turn on his video so people at home can see him.
Okay. Cool. All right. So yeah, these desert ants can count the number of steps and estimate their position relative to their nest in featureless deserts. So for path integration to um, okay, so for path integration to work, and this is something that Jim has already talked about earlier, the animal needs to know the relationship between their motion in space and the distance metric in their internal representation of space. And this conversion factor that relates these two variables is called the path integration gain. And as you might have known already, this gain needs to be finely calibrated, or else the path integrator is going to accumulate error over time. And previous work in the lab looking at the hippocampal place cells has shown that the path integration gain, as instantiated by the hippocampal place cells, is in fact highly plastic and can readily be recalibrated upon visual feedback. However, the hippocampal place cells are only one part of the navigation circuit, and it is still unclear how exactly the navigation circuit as a whole orchestrates this computation. So in this study, we focused on head direction cells, which are cells that are directionally tuned towards the animal's heading direction. And these cells are thought to provide the directional input that locks the internal map to the external world. So specifically, we looked into a thalamic nucleus, the anterodorsal nucleus of the thalamus, or ADN for short, and simultaneously recorded from hippocampal place cells and ADN head direction cells to see how the navigation circuit orchestrates the path integration recalibration computation. And this study was carried on together with a postdoctoral fellow, Ravi Krishnan Jayakumar, in the lab. Okay, so the setting we used was the dome, which Jim already explained, so I'm not going to go over the details too much, but rather keep it concise. But this is a virtual reality system, just like a planetarium, where the animals on the circular track will circumnavigate in pursuit of food and keep making laps in this in counterclockwise manner. And then there are visual landmark cues that are being projected on the inner surface of the wall. And these visual landmarks will move in the same direction as the animal or in the opposite direction as the animal, so as to create conflict between the visual landmark-based position and the path integration-based position. Okay, and again, this is a schematic or an animation of how things work inside the dome. The visual landmarks are controlled by a gain factor, a visual gain or experimental gain factor, which relates the animal's velocity in landmark frame relative to the animal's velocity in lab frame. And here the landmark frame, I mean, by landmark frame, I mean the animal's perceived speed relative to these moving landmarks. So on the left, you're going to see the animal running around and the visual landmarks moving. And on the right, you're going to see the animal's perceptive speed in the lab frame as well as relative to the moving landmark frame. So when the gain is greater than 1, the visual landmarks will move in the opposite direction as the animal, which creates the illusion as if the animal is moving faster than they actually are. Okay. And the converse is true for a gain less than one condition. In this case, now the visual landmarks will move in the same direction as the animal. So if you look at the landmark frame of reference, you see that the animal's perceptual speed is slower than its actual speed. So by using different types of gain, we can control the exact amount of conflict that animals receiving between vision and path integration. And we can see what happens to its path integration computation. Okay, so a typical experiment consists of 4 sessions or 4 epochs. The first epoch, we keep the landmark stationary, which is a gain of 1 condition. And then we slowly ramp up or down the gain until it reaches a target value G final. And in epoch 3, we keep the value steady. Okay, and the critical epoch is this epoch 4 when we turn off the landmarks. And in epoch 4, because there's no landmarks, the animal has to navigate the virtual reality solely using its path integration cues. And now the question is, now that the animal has gone through this persistent conflict of visual feedback and its path integration cues, what happens to its path integration computation in epoch 4? Okay. So first, we focus our analysis on epochs 1 through 3 when visual landmarks are present. So on the top, I'm showing the experimental gain profile where we start the gain of 1, where visual landmarks are stationary, and ramp it up the gain to a value of 1.35. And on the bottom, I'm showing a spiking plot of 2 example cells that were simultaneously recorded in this session. The blue is a place cell, and the red is the head direction cell. Okay, so just to orient people, the graph, the x-axis is the number of laps in the lap frame, and the y-axis is the location of the animal when the spiking occurred. For place cells, I'm plotting where the animal's body position was in the circular track. And for head direction cells, I'm plotting the animal's heading direction in which spiking was observed. Okay, so when the landmarks were stationary, we see that both place cells and head direction cells fire consistently at the same location every lap. For place cells, it fires around 120 degrees, and for head direction cells, it fires when the animal's facing towards around 200 degrees. However, once we start moving the visual landmarks, the firing starts happening all over the place, and this keeps happening in epoch 3 as well, as if the cells lost their tuning. Nonetheless, when we look at the firing in in landmark frame, that is the firing direction or position relative to the moving landmark frame, we see that the tuning is maintained and that the cells are firing at the exact same location or direction relative to the landmarks. So this suggests a strong control of landmarks. And to quantify this effect, we computed what we call the CA1 gain or the head direction gain. So for CA1 gain, we used all the place cells that were recorded in this session and computed a gain factor, which is how much do the hippocampal place cell representation update itself relative to the animal's motion in the lab frame. And when we compute this value, we see that this hippocampal gain perfectly lands on top of the experimental gain that we impose. And similarly, when we compute the head direction gain, we see that again, this gain value lies perfectly on top of the experimental gain, suggesting a strong control of landmarks on the firing of the internal representation of space and direction. Okay. And to make this point even clearer, we plotted the activity of head direction cells as a polar plot. So here, I'm plotting all the head direction cells that were recorded from the specific animal, where different color indicates different cells. And in epoch 3, when the landmarks are moving, we see that there's pretty much no tuning in the lab frame. But when we look at it in the landmark frame, we recover a nice tuning. Now that we established the strong control of landmarks for both place cells and head direction cells, our next question was, what happens in epoch 4 when we turn off the landmarks? Okay. And one possible scenario is that there is no learning happening, in which case the cells will start firing once per lap again, just like in epoch 1. Another scenario is that the animal fully learned its new association between self-motion and its gain of 1.5, and that the cells will keep firing even after we turn off the landmarks, as if there are visual landmarks rotating together with the animal. And the answer is neither. And in fact, what we see is that when we decode the gain value from both place cells and head direction cells, they take an intermediate value of around 1.25 in this specific session. But the important thing here is that, as you can see, the blue trace and the red trace are, again, overlaying on top of each other, suggesting that the path integration gain that is being that has been exhibited by these two systems is still coupled despite strong landmark Q. Okay? And to make sure that this gain value that the system took was in fact related to the previously experienced visual gain instead of the system randomly picking this value for no reason, we repeated this experiment with different experimental gain values. So on this plot, I'm showing for each color indicates different animals. And when we plot the CA1 gain in epoch 4 against the previously experienced experimental gain, we recover this nice linear relationship, suggesting that in fact, the epoch 4 gain that the system exhibits is predicted by its previously experienced visual gain. And moreover, the slope is less than 1, suggesting a partial recalibration where the system did not fully recalibrate to the visual gain that it experienced. So this is a nice reproduction of the previous CA1 study. But the question is, then what happens to this the head direction gain? Here, we plot the head direction gain in epoch 4 relative to the epoch 4 CA1 gain. And we see that all the points lie along the y equals x axis. This suggests, in fact, that the two systems are strongly coupled under this gain manipulation. So this is a nice confirmation that, okay, the two systems are coupled. But the question is then, where exactly does recalibration happen? One scenario you can think of is that the recalibration happens in the head direction ring or upstream of it, perhaps in the angular head velocity inputs, and that the place cells inherit this gain. This is a feed-forward model. Another scenario, though, is that actually the head direction ring might not have been recalibrated, but rather the recalibration took place downstream of the head direction cells in the place system. And by place system, I mean place cells or grid cells or other spatially modulated cells. And also perhaps in the linear velocity inputs to these cells. And that through feedback coupling from the place system to the head direction ring, the head direction ring exhibited this recalibration phenomenon. And so far, these two possibilities cannot be dissociated because we're looking at locomotor periods when the animal is running around in a circular track, and the body position and its head angle are tightly coupled. So to dissociate these two possibilities, we turn to head scanning behaviors. So head scanning is a behavior in which animals make lateral head motions with minimal body motions. And as shown in this left schematic, and when we track the animal's head position using an overhead camera, we in fact see these head scanning events happening inside the dome. So the blue trace here is when the animal's running around in a circular lap, and you see this blue circle. But we also sometimes see this red blip that happens. And this is when the animal pauses for a second and looks around the environment, scanning what's happening inside. So what we did was to extract these head scanning behavior moments and decoded the gain only using spiking activity that happened during head scans. Okay, and what we get is this, where when we plot the head direction gain during head scans relative to the previously experienced experimental gain, we see no correlation. And similarly, when we plot the head direction gain during epoch 4 when the animal's head scanning versus when the animal's running, we again see no correlation. And in fact, the head scanning gain takes values that are pretty close to 1, as if there was no recalibration in the system. Okay, so this sort of points to a scenario in which in epoch 4, when the animal's running, the head direction ring has a specific gain, but then once it pauses and looks around, it instead exhibits a gain of 1 instead of the recalibrated gain during locomotion. So going back to the two models that we used to set up the question, the finding suggests either the feedback model in which the recalibrated gain exists in the place system, or alternatively that the head direction ring learned to exhibit two different gains depending on the behavior that the animal is taking. But regardless of the interpretation, the finding rejects the simple feed-forward model of recalibration. And together, we see that the place cell system and the head direction system exhibit coherent recalibration during locomotion, albeit lack of recalibration during head scans. And with this, we demonstrate the plasticity mechanism in the navigation circuit that keeps the internal head direction and the position in register with the external world. With that, I'd like to thank everyone in the Cunium Lab and the Common Lab, as well as all the funding agencies. Thank you so much.
Okay, we have lots of time for questions. again? Or we could regress.
So, so you have these two, as I say, two sort of motion modes. One where the rat is sort of running, so that's what we recalibrate it because of that's where you play the game. The other one, you move your neck around. Yep. That wasn't. So I don't know if you tried or you're thinking, what would happen if you change the game, upon the neck movement of the rat, then you.
Yeah. Yeah, so the question was, during head scans, the animal is moving its neck. So what happens if we actually impose a gain when the animal's neck is moving? And yeah, that's a good question. And in fact, we were thinking of it as a next question of whether if we put the animal in the mid center of the dome and then let only the animal move its neck and only apply the gain, and then put the animal back on the circular track, do the place cells exhibit recalibration, even though it never experienced the locomotor gain? That's actually a follow-up question we were actually thinking of. Yeah.
You mentioned that they do this head scan when they're paused.
Mhm.
So what do the place cells do when they're paused? Are the place cells active, and do they represent accurately the position in the environment?
Yeah, so we didn't analyze the place activity, but my guess is that as the animal's looking around, it's going to have 4 theta sequences in which instead of the representation locked to its position, it's going to sweep to its future direction that it's looking towards. And so my guess is that probably there's some non-local coding happening during those moments.
They mostly stay firing at the location where the rat is. Then we didn't look carefully at any forward coding or not, but when the animal is standing there doing head scanning, the place cell is just firing, is the simple answer.
Yep.
Just curious, during the different epochs, when you change the game, is there any head scanning behavior difference at different epochs? Or would that correlate with the game increase, decrease on?
Yeah, so the question was whether the head scanning behavior changes between epochs. So we did not really analyze it carefully, but we didn't see a difference related to gain, but we did see an increase in the frequency of head scans towards the end of the session, like in epoch 3 or epoch 4. And perhaps it's because initially the animal's hungry, so it's going to keep running and running in pursuit of food. But as it gets satiated, perhaps it starts looking more carefully, and yeah.
In terms of in your session, the game doesn't match the most recent experience, and I'm wondering if you guys think that that's a product of how much experience the animal had there, or do you think it's something adaptive about not having it back, something less than what amount of experience?
Yeah, so the question is, why did the system not perfectly recalibrate, but rather maintain an intermediate value? And yeah, my guess is that if we keep the epoch 3 length longer, such that the animal keeps this visual gain, then perhaps the system will get closer and closer to the actual visual gain.